Hello, my dear students, and welcome back. I know your board exams are so close and you're all working incredibly hard. So today, let's do a super quick and super easy revision of a very important chapter, Recombinant DNA technology. Chalo, let's get you those top marks. Okay, let's start with something fun. You all love those amazing song remixes you see in Instagram Reels and on YouTube, right? You know, where a DJ takes a bit from one song and mixes it into another to create something completely new and awesome? Well, what if I told you scientists can do the exact same thing, but with the song of life itself, with DNA? So first things first, let's get the big idea down. What exactly is this DNA remix? This is the foundation for the whole chapter beta, so listen very carefully. Right, so in your textbook, it's called Recombinant DNA Technology, or rDNA for short, but you've probably also heard of it by its more famous name, genetic engineering. And really, it's just that, methods for directly changing the DNA of an organism. It's like being a DJ, but for genes. The whole idea is to take one useful gene, let's say a gene that makes a firefly glow, and put it into a completely different organism. And what happens? Well, that organism gets a brand new amazing ability, a superpower. It's that simple, and it's absolutely fascinating. Now, to pull off this amazing genetic remix, our scientist DJs need a special set of tools. It's not a hammer or a screwdriver, of course. They have a very cool molecular toolkit. Let's open it up and see what's inside. Okay, this slide is super important for your exam, bacha. You need 4 main tools. First, restriction enzymes. Just think of them as tiny molecular ganchi or scissors that cut DNA at very specific spots. Second, DNA ligase. This is our genetic favicol, the glue that sticks the DNA pieces back together. Third, a vector. This is our delivery van, which is usually a small DNA circle called a plasmid. It's what carries our gene into the new cell. And finally, of course, the gene of interest. That's the superpower gene we actually want to copy. Now for the part that you'll definitely see in your 5-mark questions. The actual process. How do all these tools work together? Well, you can think of it just like cut, copy, and paste on a computer, but for DNA. Let's go through it step by step. So, step 1, we need 2 things to start. We have the source DNA, maybe from a human cell, that has the superpower gene we want. And then we have our delivery van, the plasmid. Plasmids are just these tiny circular pieces of DNA found in bacteria, and they are perfect for this job. Okay. Next up, we bring in our molecular kink sheet, the restriction enzyme. Now here's the most important part. The same enzyme is used to cut both. It snips out our gene from the human DNA and it cuts open the circular plasmid, creating the perfect docking spot for our new gene. In step 3, our superpower gene just naturally slots into that open space in the plasmid. The ends of the gene and the plasmid are sticky, you see, so they are naturally attracted to each other and they join up. It's a perfect match. And for the final step, we bring in our genetic favicol, the DNA ligase enzyme. It works like a superglue, permanently sealing the gene into the plasmid. And ta-da! We've done it! This new remixed plasmid with DNA from 2 different places is what we call recombinant DNA. Now listen up. This is a 1-mark question just waiting to happen. You absolutely must remember these 2 names, Stanley Cohen and Herbert Boyer. In 1973, they were the brilliant scientists who first performed this experiment successfully. They are the fathers of genetic engineering. Don't you forget it. So, we know how it's done. But so what? Why is this so important? Well, let me tell you. This technology hasn't just been useful. It has completely changed our world. Let's look at some real-world superpowers it has created. Take insulin, for example. Scores of people with diabetes need it to live. Before, we had to get it from the pancreas of pigs and cows. It was super expensive and sometimes caused allergies. But now, thanks to rDNA, we just put the human insulin gene into bacteria. These bacteria become tiny insulin factories, producing huge amounts of pure, safe, and affordable human insulin, called Humulin. What a game changer. And the breakthroughs just kept on coming. In 1982, we got Humulin. Then 1996 was a massive year. We got BT cotton, a crop that creates its own pest shield, saving our farmers lakhs of rupees. And we also got Dolly the sheep, the very first cloned mammal. And more recently, the COVID-19 vaccines that were developed in record time. Yep, that was also possible because of this amazing technology. And the list just goes on and on. We have growth hormones, medicines to dissolve blood clots,
safer vaccines for hepatitis B. In farming, we have golden rice with extra vitamin A and crops that can survive with less water. And in forensics, we have DNA fingerprinting to help solve crimes. It's literally everywhere.
Chalo, I think you've got the concepts down perfectly. So now, it's time for a quick fire question round. This is just to check your knowledge and get you all set for those objective questions in the exam. Are you ready? Let's go. All right, here's your question. It's a classic one. Who discovered the plasmid? Remember our little delivery van. Is the answer A, Paul Berg, B, Sir Alec Jeffreys, C, Joshua Lederberg, or D, Carey Mullis? Take a second, think about it. And the correct answer is C, Joshua Lederberg. If you got that right, shabash, you are absolutely correct. He discovered these amazing little tools all the way back in 1952. You are definitely on track to ace this exam. So, if you need to remember just one sentence to summarize this entire chapter for an answer, this is it. The fundamental theme of RNA technology is the isolation and propagation of a desired DNA molecule. That's all it is. You find a gene you want, you take it out, and you make tons and tons of copies of it. Simple. So think about this. For the first time ever, we humans are not just reading the code of life, we are actually learning how to write it. We can design life to help us solve the biggest problems. So my final question to you is, if you were the genetic engineer, what superpower would you choose to create next? Give it a thought. All the very best for your exams, my dear students. I know you will do wonderfully.